Sorry, some network issues. Sorry. Okay, let us start the class. Okay, so student, in the last class we have studied about the applications of biotechnology, right? So in that I have left the ethics. So before going to start the class, so here is the question for you. Answer me for my question. Okay, the question is biomolecules are. Okay, so they are the inorganic material only. Option B, they are the organic material only. Option C, all the carbon compounds obtained from the living tissue. Yeah, and from the living tissue. So, and option D is only DNA and RNA. Can you answer to this question, student? Can you answer me for this question? Which is the correct option? It is the correct option. Answer me in the chat box. Can you tell me whether I'm audible and screen is visible first? Can you tell me in the chat box or in the poll whether I'm audible and screen is visible? So please, student, answer me in the chat box. Jainvi Chitradurga, admin. Can anyone tell me? Okay. So thank you. Let us start the class. So biomolecules, right? So the correct option is option C, all the carbon compounds obtained from the living tissue. Okay. So let us study in detail about the biomolecules today in our class. Okay. So in the last class, I have not told you the ethics in the biotechnology and the biopiracy. Right, biopiracy. Let us study about the ethics. What is the ethics? Okay, genetically modified organism, it should not harm the society, right? It should not harm the society. So that's the reason. So it should not go against the human morality. Okay, humans morality. Okay, so that's why. So the any product or any genetically modified organism form means, so they have to check by the GEAC. Okay, what do you mean by GEAC? Can anyone tell me? GEAC? They have to take a permission from genetic engineering approval committee. Okay, genetic engineering approval committee. Okay, so they have to take a permission from them. Okay, so it will check whether that organism or which organism or any product which is formed by the genetic engineering, whether it is harmful to the human beings or useful. Okay, the only useful products will be released. Okay, only useful products will be released in the markets. Okay, so that is what we will call it as the ethics. Okay, for example, if you see any humans are created, okay, by humans are created by modifying his gene. Okay, so is behaving like a some differently. Okay, or he's having some different diseases or some new diseases will come, right? So on that type, it should be harmful to the society only, right? By modifying the gene, here we have introduced the different disease into the world, right? So that things we should not be happen, right? So that's why. So whenever any modified organism or anything formed in the genetic engineering means, they are going to check or they are going to get a approval by the genetic engineering approval committee. So then only it will be marketed. Okay, so that is the GEAC. Okay, so then coming to the piracy, biopiracy. What do you mean by biopiracy? Means sometime what will happen, some countries or some company, what it will do, they will take the resource of the other country or the products of the some company, and without informing that country or without informing that company or giving the money to them. Okay, they have to give the money, right? They will not give anything. They are not giving even the name also. Okay, they will utilize that bio resource and they will give their name. Okay, they will give some new name and they will utilize that. Okay, so that is what we will call it as the biopiracy. Okay, so for that's why for right, we will call it as a patent. So patent will be given for only for the product. So you are by utilizing your experiments and something you have did, and you have created one product. Okay, that product should be sold by you only. So that government will give the patent. Okay, the government will give the patent to you. The in the name you have given some name, right? For example, you give some names to that product. That product should be sold under that name only, under prescription of yours. Okay, with their by taking the permission from you, and they have to sell that. So that is what we will call it as a patent. Okay, so the no other person should not synthesize the same thing what you have synthesized by utilizing the same thing, and they should not sell. For example, you are created one tablets or something. Okay, so now you are selling that in the 100 rupees. So the another company, what it will do, that will create that same tablet and they will sold it in the 50 rupees. Okay, then the people will take the 50 rupees tablet, not the 100 rupees tablet, right? So then your 10 years of the experiment become loss, right? So this is what, for this kind of things, they are given the patent. Okay, so patents are given for the product. And Copyright Act. Copyright. Okay, Copyright Act. Copyright Act will given for the writings. Okay, for example, you have written, you have did your PhD. Okay, so you have written some principle, you have written some project works or some theories and something you have written. Okay, so that should be copied by the another person, and he can remove your name and he can give his name in that, right? So it should not be done. Okay, so for that writing, they will give the copyright. So if you see some books and if you see some details, they will give the Copyright Act, right? So this is what we will call it as a Copyright Act. Okay, Copyright Act will given for the writing. And when it comes to the biopiracy, we will remember that our basmati rice, right? So India, from the so many years, we are growing the basmati rice. Okay, but in the 1997, what happened? One of the American company came. Okay, so one of the American company came and he's synthesized. He is growing the basmati rice there and it is selling there. Okay, so but before that, the India is used to give the rice. But when this American company started doing that, so this no one is purchasing the rice from the India. But in the 1997, what happened? This American company take the patent on the basmati rice. Okay, but that is a resource of the India, right? But they have taken the patent, right? So then in the court and something cases has happened, but finally came to know that the basmati rice was grown in India from the so many years. And here we are growing the 27 varieties, and we are having the 2 lakh varieties of the basmati rice. Okay, so that is what the American company canceled the patent on the basmati rice. Okay, so not only based on that, so you can take the our turmeric and neem, right? So turmeric and neem. Okay, so we know that from the so many years, right? So over so many years, our ancestors, they are using the turmeric and neem because of this antibiotic property, right? But here what happened? Some companies try to take the patent on Ayurvedic property and the medical property and also the antibiotic property on the turmeric, but that is also failed. And based on the neem also, it was failed. Okay, because then finally it is decided that there is no patent for the herbal product. Okay, so that's why they're not giving the patent for the bio herbal product. Okay, so no patent for herbal products. Okay, fine. So this is about the ethics and this is about the biopiracy. Okay, so obviously whenever the that company or whenever the other countries is using our resource, means they have to give compensation for us, right? Without giving those all the things, simply they are taking the resource and they're utilizing, means it is not a good thing, right? So that's why this biopiracy came. Fine. So here is another question for you, student. So answer me, which enzyme was first isolated and purified in the form of crystal? In the form of crystal. Can anyone answer me? Which is the answer? I will not run the poll. You can answer me in the chat box. Okay. So you can answer me in the chat box. Yes. So the correct answer is urease, right? Yes, the correct answer is urease enzyme. Okay, urease enzyme is the first enzyme to be purified in the form of crystal. Okay, in the form of crystal. Okay, so let us go to the biomolecules. So student, just we are revising the biomolecules. Okay, so let us see what is the biomolecule. Everyone know that, right? What is the biomolecule? So biomolecule is, so for example, organism, living organism. So I am a living organism, you are a living organism. In our all the body, there is a biomolecule is present, right? So which are the biomolecules, student? Can anyone tell me? Can I tell carbohydrate? Can I tell carbohydrate, student? Carbohydrate, protein, and lipids, and nucleic acids, right? So these all the things will come under the biomolecules. Let us study. So biomolecules are the chemical compounds found in living organisms, right? They are the chemical compounds which are present in living organism. So it may be, it include organic also and the inorganic compound also. Where you can see the inorganic compound, organic compound, so you know that, right? Carbon and the hydrogen you can see, you can tell. So when it comes to the inorganic compound, can you see phosphate group in the DNA? Right. Then can you see nitrogen in the protein? Right. So these all are the inorganic compounds. Okay. So let us go to this thing. Okay. So before going to start, so first we will learn about what is the metabolites. Okay. What is metabolites? So student, you know what is the metabolism? You know what is the metabolism? There are two types of the metabolism, right? Can anyone tell me what are the two types, student? Can anyone tell me? Yes. Anabolism and catabolism. Right? Anabolism and the catabolism. So just remember that what is anabolism? Anabolism means constructing work. Okay, constructing. In our body, the daily protein is going to be synthesized, right? From the amino acid, from the smaller molecule, from the amino acid, the whole protein is going to be synthesized, right? So that is what we will call it as a anabolism. Anabolism is nothing but constructive work. Okay. Just remember how the building construction will done by using the bricks, right? So here also they are going to construct by using the things, by using the amino acid, by using the simpler molecules, right? So this is what we'll call it as a, this is constructive, okay? Constructive. For example, you can take the, you know, in the photosynthesis, what will happen? By utilizing the carbon dioxide and the hydrogen, the plant will synthesize the carbohydrate, right? So this is what the example of the anabolism, okay? Or you can take the glycogen synthesis. In the glycogen synthesis, what will happen? The smaller glucose molecule will combine together to form the glycogen, right? So you can see here, photosynthesis. Photosynthesis. By utilizing carbon dioxide and water, it is going to give the C6H12O6, right? So that is called it as the anabolism. You know that for the anabolism, if we are constructing the home means, we require money, right? Yes. In the anabolism also, we require money, but that is not a money which we will use outside, okay? Which we will use inside our body. What is that energy, student? ATP, right? Yes. It requires the ATP. So that is energy currency, right? It requires the energy currency. So when it comes to the catabolism, catabolism is destructive. Okay, so it is the destructive reaction. For example, you can take the glucose. Okay, so glucose is broken down by the glycolysis. Can you recall, student, by the glycolysis, it can be broken down into the pyruvate and 1 minute. Okay, so this is about the glucose. So you know that, right? During the glycolysis, what will happen? So by utilizing the oxygen, we will broken down the glucose into the pyruvate, right? So glucose into the pyruvate, right, student? Glucose into pyruvate plus ATP is released, right? So here only the tricks is there, right? So anabolism means construction, we require money, right? When it's a catabolism, it is a destruction. It does not require the money, but it will give the money. Okay, it will give the ATP, energy molecule. Okay, so let us talk about the metabolism. So student, there are two types. One is a 1 minute. So you know what is the primary metabolites? Students, you know what is the primary metabolites? Primary metabolites. What is the primary metabolites in the body? So student, these primary metabolites are very important. So it will be helpful for the growth. It will helpful for the growth, reproduction, and survival of the organism. Okay, survival of organism. Okay, so this is required for growth of the organism. For if the organism is reproducing, means for that required, and for the survival of the organism. It is a very important. So then which are the things it will come? You can take the carbohydrate, you can take a protein, you can take a lipids, and you can take a nucleic acid. Okay. So these all the things will come under the primary metabolites. Okay. So let us talk about the secondary metabolites. Secondary metabolites. You know, right? What is the secondary metabolites? You know, right, student? Can you tell me? Can you recall what is the secondary metabolite? Is that going to involve in the growth and reproduction and survival of the organism? No. Okay. It is not involved in growth. It is not involved in growth, reproduction, and survival. Then why it is required? Secondary metabolite is required for defense mechanism. Defense mechanism. Okay. So see here, primary metabolites, secondary metabolite. Primary metabolites is synthesized by all the organisms, including the animals. It is going to be synthesized. When it comes to the secondary metabolites, it is going to be synthesized from only plants and only fungi and only bacteria. Okay. So it is not at all synthesized by the animals. Okay. It is not synthesized by the animals. 1 minute. Okay, sorry, student. Okay, so this is about the primary metabolites and the secondary metabolites. Secondary metabolites is not at all synthesized by the animals. Okay, so secondary metabolite, the animals will take from the plants. Okay, so this is the secondary metabolites. So they will secrete the secondary metabolites for the defense mechanism. For example, some microbes came and attacked the plant or fungi. Okay, so then it started to releasing the secondary metabolite. Okay, so that is what call it as a defense mechanism. It is not synthesized by animal. Okay, so this is you have to remember secondary metabolites. Okay, student. So here they are given the some secondary metabolites. Okay, so just we'll go through that. In the exam, it is very important. You have to remember these points. Okay, so pigment. Let us come to the pigments. These pigments are secondary metabolites. Carotenoids, alkaloids. Alkaloids, where you studied alkaloids in the alkaloids, can anyone tell me? Drugs and alcohol abuse. In the drugs and alcohol abuse, you study about the morphine, right? Morphine. So from the poppy plant, the morphine will be obtained, right? Poppy plant, from the latex of the poppy plant, right? In the health and human disease. So, and the codeine. Codeine is also the type of the morphine only. Okay. So both are used as the pain reliever. Okay. They are used in the pain reliever. Okay. Pain reliever. For the analgesic, they will use. Okay. So then coming to the terpenoid, monoterpene and the diterpene. Okay. So essential oil, when you take a lemon in your hand, so the oil will be released, right? So oil will be released and you will get the smell of the lemon, right? So that is also the one of the secondary metabolite. And abrine and the resin, they are the toxin. And concovalin is, when it comes to the lectin, they are glucose binding protein. Okay, concovalin is glucose binding protein. Okay. So this concovalin is used in the separation of or the purification of carbohydrate in the mixture. For example, the mixture is consisting of the carbohydrate, protein, and lipid. Now I want carbohydrate to be removed, right? Now I want the carbohydrate to be purified. On that time, what I will do, I will add the concovalin. Concovalin will attach to all the carbohydrate. Then I know a method how to remove the concovalin. Then I will remove the concovalin. From the concovalin, I will remove our carbohydrate. Okay. So then coming to the drug, so vinblastine and the curcumin. So here you can see vinblastine is given in the cancer treatment. Okay. It is given in the cancer treatment. So vinblastine and vincristine. So these all are the secondary metabolites. Then coming to the polymeric substances, rubber, gums, and cellulose. Okay. So these all are the secondary metabolites. You have to remember this. Okay. If you have any confusion here, student, can anyone tell me if you have any confusion here? If you don't have, means let us go to the next. Okay. So then coming to the biomolecule. So whenever we are talking about the biomolecule, so there are two types will come. Okay. Biomolecule, two types. One is the macromolecule and another one is the, 1 minute. Biomolecules. Here there are two types, student. Okay. So here two types. One, I will call it as the macro. One is called it as the macromolecule. And another one I will call it as the micromolecule. Micromolecules. Okay. So which are the example for the macromolecule? Macromolecule should have higher than the 100 Dalton. Okay, 100 Dalton. When it comes to the micromolecule, they're having the less than 1,000 Dalton. Sorry, not 100, 1,000 Dalton. Okay. So macro means there are the molecules, they're having more than 1,000 Dalton. That is the size. Okay. When it comes to the micro, they're having the less than 1,000 Dalton. Can you give me the example for macromolecule? If you take protein, right? If you take the polysaccharide, and if you take the example of, yeah, can you give me the example of macromolecule, DNA, RNA, and lipids, right? So these all the things will come under the macromolecules. Okay. So let us see here. Let us talk about the micromolecules. Which are the micromolecules are there? So protein is made up of which molecules? Can anyone tell me? Yeah. So it is made up of amino acid, right? It is made up of amino acid. Yes. So our amino acid is having the less than 1,000 kilodalton. So that's why we will call it as it is a micromolecule. So then coming to the polysaccharide. Polysaccharide is made up of the glucose unit, right? Yes. So glucose unit, that is also the micromolecule. And coming to the nucleotides. Okay. So this is also the micromolecule. So, but here you can see our lipid is only 800 Dalton. Okay. 800 Dalton. But why we are keeping that in the macromolecules? Can anyone tell me? Why we are keeping that in the macromolecules? Why not we are keeping in the micromolecules? Can anyone answer me? Why not we are keeping that in the micromolecules? Yes. So what is the reason, student? Yes. So see here, the lipids is insoluble in the acid. Okay. So that is the reason. So we are keeping that in the lipids, that we are keeping in the macromolecules. Okay. Yes. So yeah. So then coming to the next, carbohydrate. Okay. So student, can anyone tell me? So what is why we will call it as a carbohydrate or hydrates? You can tell me. Remember here, carbohydrate is made up of three things, right? One is a carbon, one is a hydrogen, and another one is a oxygen, right? Student, just recall. So what is the component of the hydrogen? Sorry, what is the component of water? H2O, right? Simple. So in the carbohydrate, what is the general formula? CN H2N ON, right? See here, I will write the glucose molecular formula here, C6H12O6. Okay. So here you can see here, hydrogen to oxygen ratio is H12O6, right? 1 and 2, right? So that is equal to the water, right? So equal to the water. So the the oxygen and hydrogen constant ratio is same as in the water. So that's why they will call as the hydrates of carbon. Okay. So ratio of hydrogen and oxygen is same as in the water, same as in the water. Okay. So that's why we will call it as, that's 1 minute. So that's why we will call it as the hydrates of carbon. Okay. Hydrates of carbon. Okay. So student, do you know the structure of the glucose, how it will be? It is consisting of so many hydroxyl group and it is consisting of the aldehyde group, right? Yes. So what is the full form? What is the full form of the what can we give the definition for the carbohydrates then? Can anyone try? Yes. So we'll call it as polyhydroxy ketone or aldehyde. Ketone or aldehyde. Okay. Polyhydroxy ketone or aldehyde. Okay. So there's a definition of carbohydrate. Okay. So let us talk about the carbohydrate. Carbohydrates will also call as the saccharide. Saccharide. Okay. So saccharide means what? Sugar. Sugar. So it is not compulsory that all the carbohydrates should be sugar. Okay. For example, we will eat the rice, right? So is that rice is more of sugar? No. Right? Yes. So not all the carbohydrate molecules should be a sweet in taste. For example, cellulose. Cellulose is made up of the carbohydrate, glucose, right? Is that sweet in taste? No, right? Polysaccharides will not be sweet in taste. Okay, only the monosaccharide will be sweet in taste. Okay, yeah. So that's why we will call it as the saccharide. Then coming to the classification of carbohydrate, there are 3 types. I think you know this, right? One is the monosaccharide. One is the monosaccharide, and second one is the oligosaccharide, and third one is the polysaccharide. Okay, third one is the polysaccharide. So monosaccharide means what? It is consisting of the single sugar unit, right? It is consisting of the single sugar. We will call it as the monosaccharide. So if it is consisting of the 2 to 8 molecule, we will call it as the oligosaccharide. It is consisting of more than 8 means we will call it as the polysaccharide. Okay. Yes. So can you give me the example of monosaccharide, student? Can you give me the example of monosaccharide? Glucose, fructose, and galactose, and mannose, and raffinose. These all the things will come under the monosaccharide, means it is having only one sugar. Okay, one sugar molecule. So then coming to the oligosaccharide, so here you can call the disaccharide. Which are the disaccharide? Can you tell me? Maltose, and can I tell lactose, student? So student, lactose is the milk sugar. Okay. So whenever your mother give you sprouts, right? Or in the school, they will give the sprouts, right? This sprout is made up of the sugar that is a maltose. That's why we'll call it as the malt sugar. Maltose is the malt sugar. And we can talk about the sucrose. Sucrose, what is the sucrose here? Sucrose is the disaccharide, and sucrose we will call it as the cane sugar. Cane sugar. Okay. So we can give me some other examples also. Okay, student. So now let us come to the polysaccharide. So polysaccharide is made up of more than 8 sugar molecule. So that's why we'll call it as a polysaccharide. Okay. So let us come to the polysaccharide. There will be a homopolysaccharide. Homopolysaccharide, which is consisting of same glucose unit, same sugar unit, or heteropolysaccharide, which is consisting of different, it is consisting of different sugar unit. Okay. So let us talk in the next. Okay. This is about the sugar, carbohydrate. It is a polyhydroxy ketone and aldehyde. It is a saccharide. We will call it as a saccharide because it is sweet in taste, but not all the carbohydrate will be sweet in taste. Okay. Okay. So let us go to the next. So here you can see the open structure. Okay, open structure of the sugars. You can see the aldose here. So student, here you have to remember what is the difference between glucose and the fructose. Can anyone tell me what is the difference between the glucose and fructose? Glucose and fructose. Both of them having the molecular formula C6H12O6. Okay. So, but when come to the glucose, it is the aldehyde, and when come to the fructose, it is a ketone. Okay. Ketone sugar is the fructose. When it comes to the aldose sugar, that is the glucose. Okay. So here you can see the glyceraldehyde structure and dihydroxyacetone and glyceraldehyde. Ribose we can see in the RNA and the glucose structure. See here, why I have told you polyhydroxy. See here, there is a different number of the hydroxyl group is there, right? So that's why we will call it as a polyhydroxy, either ketone or aldehyde. Okay. Okay, see here, this is the closed structure, or we can call it as a ring structure. Here you can see there is a difference between glucose and the fructose. So fructose is having the furanose ring, right? Fructose is having the furanose ring, whereas the glucose is having the pyranose ring, right? Pyranose ring, right? So you can remember like this. Let us go to the disaccharide. Disaccharide, I have given the example to you, right? Lactose, right? See here. Lactose. 1 minute. So let us talk about the lactose. Lactose is made up of glucose and galactose, right? Galactose. When it comes to the maltose, maltose is made up of, can anyone tell me? Glucose plus glucose, right? Can anyone tell me sucrose? Sucrose, student? Sucrose is made up of glucose plus fructose. Okay, glucose plus fructose. See here what will happen. So this all the molecules is having the free hydroxyl group. Okay, this is having free OH group. Okay, free OH group. So that's why this is called as a reducing sugar. Reducing sugar. All the monosaccharides and all the disaccharides are reducing sugar. Okay, reducing sugar except sucrose. Okay, except sucrose. Sucrose is the non-reducing sugar. Why we will call it as a non-reducing sugar? See here, it does not have any free hydroxyl group. Okay, here you can see there is no free hydroxyl group here. It is having the CH2OH, right? So that's why we'll call it as the non-reducing sugar. But you see here in the lactose, it is having the HOH, right? The OH is there, free hydroxyl group. So that's why it is the reducing sugar. And when it comes to the maltose, it is also having the reducing sugar. And cellobiose, it is also having the reducing sugar. Okay. This is about the disaccharide. In this slide, you have to remember sucrose is the non-reducing sugar because it does not have any free functional group. Okay. Okay. So then coming to the polysaccharide. So what we are going to learn about the polysaccharide is, I have told you that polysaccharide is made up of the more than one unit, one sugar, right? More than 8 sugars it is having the. Polysaccharide means what? It is having more than 8 sugar unit, right? More than 8 sugar unit. Okay. So students, see here, polysaccharide are non-sugar. Okay, they are non-sugar. And one more thing you have to remember, they are not sweet in taste. They're not sweet in taste. If you take the cellulose and if you eat that, means it is not sweet in taste. Okay. So biopolysaccharides are not sweet in taste. Okay. So in the polysaccharide, there are two types. One is homopolysaccharide. Homopolysaccharide. And another one is heteropolysaccharide. Heteropolysaccharide. Polysaccharide. Okay. So student, when we are talking about the homopolysaccharide, there are two types will come. Okay. So can you recall what are those two types? One is the storage polysaccharide. Storage polysaccharide. I will write like PS. Okay. Storage polysaccharide. Which are those storage polysaccharide? Can anyone tell me? Starch and glycogen. Starch and glycogen. Right. So where is the starch will be present? It is present in the plants. Glycogen in the animal. So glucose is going to be stored as the glycogen in animal and it is stored as a starch in the plant. Okay. So in the homopolysaccharide only, you can give the another example. So that is structural polysaccharide. Structural. Which are those structural, student? Which are those structural? One is cellulose and another one is chitin. Another one is a chitin. So those are the homopolysaccharide. So see here, our chitin is made up of N-acetyl glucosamine. Glucosamine. It is the nitrogen containing compound. It is nitrogen containing compound. Okay, student. It is a nitrogen containing compound. What is that? Chitin. In the exam, they may ask you. Okay. So you have to remember that chitin is the N-acetyl glucosamine. It is consisting of the nitrogen containing compound. In the other exams, like in the old question paper, they have asked the question that what is a chitin or whether it is a homopolysaccharide or a heteropolysaccharide. Okay. It is important. Just remember. So then coming to the heteropolysaccharide, hetero means what? Homo means what? It is consisting of the similar, right? All the units are similar here. But when it comes to the heteropolysaccharide, it is consisting of the different units, right? It is consisting of the different units. Let us talk about like this. So see here. So this is one unit. Okay. So this is the another unit. Okay. So this is the one unit. This is the second unit. Right? So this is what we will call it as the heteropolysaccharide. The example for a heteropolysaccharide is, can you recall what is the cell wall of the bacteria is made up of, student? What is the cell wall of bacteria is made up of? What is the cell wall of bacteria is made up of? Peptidoglycan, right? Very good. Peptidoglycan. Okay. The cell wall is made up of peptidoglycan. This peptidoglycan is heteropolysaccharide. It is made up of NAG and NAM. You know what is a NAG and NAM, right? NAG means what? N-acetyl glucosamine. NAM means N-acetyl muramic acid. Okay. If you have confusion in the polysaccharide means, kindly tell me. In the polysaccharide, there are two types. Polysaccharides are not sweet in taste, and there are homopolysaccharide and heteropolysaccharide. Homopolysaccharides are storage, starch and glycogen, and structural are cellulose and chitin. Then coming to the heteropolysaccharide, it is example is the peptidoglycan. Okay. Okay. So let us go to the proteins. What are the proteins? So student, remember here. So protein is made up of the amino acids, right? So amino acids are called as building block of protein. Building block of protein. Okay, building block of proteins. Okay, let us study about the proteins. Okay, so before going to talk about the protein, you know, students, just imagine or just think. Okay, so which are the protein-rich foods? Are you eating the protein-rich foods? So whenever we are taking the cereals and whenever we are taking the vegetables, So these all the things is consisting of the protein. And our egg, right? Our egg is also consisting of the protein rich, right? So let us talk about the amino acid first. Okay. So student, why it is called as the amino and acid? See here, what is the structure of the amino acid? Okay. So it is made up of NH3, COOH and the R molecule. COO plus, we'll call it as a COO minus and NH3 plus. 1 minute. Okay. So this amino acid is made up of the, see, this is alpha carbon atom. Single carbon atom was attached to the NH3 plus and NH3 should become that side. Okay, 1 minute. NH3 plus, COO minus, and here is the H plus, here is the R. Okay, so there are 20 amino acids. So there are differ only in the R group. Okay, so differ only by the R group. Okay, see here student, alpha carbon atom is there, acidic group is there, amino group is there, right? So amino group, acid group. So that's why it is called as the amino acid. And you can observe here, in the amino acid, the plus sign is also there, minus sign is also there, right? So that's why we will call it as a Zwitter ion. Okay. So there are about 20 amino acid utilized in the protein synthesis. There are 20 amino acids are very important for the protein synthesis. Okay. So this is, you can study 1 minute. Okay. So see here. So when it comes to the amino acids, so they are given the 3 structures to you. Okay. So these 3 structures are important. You have to learn the structure. Okay. So one is the glycine they are given. Glycine is the simplest amino acid. Simplest amino acid. And it is the only amino acid which is achiral. Achiral. Okay. So achiral it is. Achiral means what? Chiral means all the alpha carbon atom was bounded by the different atoms. See here, alanine is having the one side ammonia, one side carboxyl group, one side hydrogen, and one side methyl group, right? So that's why we'll call it as the chiral. But when it comes to the glycine, it is a achiral because see here, in these 2 position, it is having the hydrogen, right? So it is called as a glycine is the simplest amino acid and it is a achiral. Okay. So see here, alanine is consisting of the CH3 group and serine is consisting of the alcoholic group. Okay. Just remember the structure. They may ask you in the exams. Okay. And coming to the Zwitter ion, see here, this is the amino acid. In the body, student, the amino acid is act as a buffer. Okay. See here. So here only plus is there, but here not minus, right? Minus is not there. So see here the plus is there and minus is there. So that's why we'll call it as this is a Zwitter ion. In the different pH condition, it is going to maintain the pH, it is going to remove the H plus H ion, hydrogen ion from the carboxyl group and the amino group to maintain the pH. Okay. So that's why it is called as the Zwitter. Okay. So Zwitter ion. Okay. So this is about the proteins. So when come to the proteins, so there are different amino acids are present. Okay. So here you have to remember only the nitrogen. 1 minute. Yeah. So here you have to remember students, sulfur containing amino acid. Okay. Which are the sulfur containing amino acid? Cysteine and methionine. Okay. So the disulfide bond will be formed by only the cysteine. Okay. Because it is having free SH group. Okay. Free SH group. When it comes to the methionine, it will not form because it is having the S and CH3 group. Okay. So that's why. So when it comes to the basic amino acid, arginine and the glycine is important. Okay. So you have to remember basic amino acid and also the sulfur containing amino acid. It is very, very important. So here I have given the 20 different names of the amino acid. Glycine, alanine, valine, leucine, isoleucine, aspartic acid, aspargine. I will give these notes. You can read in these notes. Okay. Okay. So student, whenever the body only synthesizing the protein means, it is not at all amino acid means it is not at all essential to take from the outside, right? For example, you're making a list for your home. What are the things you require? Okay, grocery shopping. So are you going to write the things which is the not which is already present in the home? No, right? You are going to write which is not there in your home, right? So like that, essential amino acids are which is required in the diet, when non-essential amino acids are not required in the diet because our body only synthesizing it. Okay. Yes. So essential amino acid taken in the diet. Okay. So it was synthesized in body. 1 minute. So non-essential amino acids are synthesized in the body. These are synthesized in body. Synthesized in body itself. Okay. Okay. So let us talk about the protein conformation. Very simple, students. See here. So whenever the amino acids are like this, so only it is made up of the sequence. Okay. Only it is made up of the sequence. That structure, we will call it as a primary structure. Primary structure. Okay. So it is only consisting of the sequence of amino acid. We can easily identify which amino acid is in the which position. We can easily identify amino acid position. Okay, so this is the first, and second one we will go to the secondary structure. In the secondary structure, see here. So it is there are the two types. One we will call it as the ellipse structure, and second one we will call it as the beta pleated sheet. Okay, beta pleated sheet. Okay, so alpha helix means what? See here, the structure is going to be formed like this. Okay, coiled around here, coiling of this peptide. Coiling of peptide. Single peptide is there. It is coiled around. Okay, so that is what we will call it the peptide. Sorry, helix. Okay, so for the helix, example is keratin. Keratin. So student here you have to remember all the protein which is helps in the structure. This is having the secondary structure. Okay, and example for the helix is the keratin. So then coming to the beta pleated sheet, see here, beta pleated sheet will be arranged like this, like the sheets. Then there will be the hydrogen bond present here. Okay, so this is what we will call it as a beta pleated sheet. And the example for the beta pleated sheet is fibroin. Fibroin. Okay, you can see that fibroin in the silk. Silk. Okay, so this is about the secondary structure. So in the tertiary structure, what will happen? The amino acid will coil around, and you can see it will form some globular structure. Okay, so it will form the globular structure like the thread. If you take the woolen thread, how it will be? Like, so like it will coil around like woolen thread. Okay, so this is what we will call it as a tertiary structure, or we can call it as 3D structure. 3D structure. Student, you have to remember here, the protein is functional in the, or the enzyme is functional only in the tertiary structure, not in any secondary structure or the primary structure. Okay, the enzyme will become active only when it comes to the tertiary structure. Okay, so here the mainly force involved in making that thread-like structure or making like that globular structure is two bonds. One is the non-covalent bond, so that is a weak bond, and the covalent bond, Okay, so these two things are involved in the tertiary structure. Okay, the example for the tertiary structure is myoglobin. Myoglobin. So what is the function of myoglobin, student? What is the function of myoglobin? Storage of oxygen. Okay, it will store the oxygen. Let us come to the quaternary structure. Quaternary structure. In the quaternary structure, what will happen? So here, the two or more polypeptide chain come together to form the one protein structure. What will happen here? Two or more polypeptide. Two or more polypeptide will come and it will join. Okay, two or more peptide will come only in the quaternary structure. Still, tertiary structure, only single peptide is there. Okay, single peptide is there. So in the quaternary structure, two or more polypeptide together, they will join and form the structure. So here also the non-covalent bond is involved, non-covalent, and also the covalent and the disulfide bond, right? and the disulfide bond is also present. Okay. So see here we are taking the example of hemoglobin. Here we are taking the example of hemoglobin. Hemoglobin is made up of 4 chains. It is made up of 4 polypeptide chains. In that, 2 alpha chain and 2 beta chain. Okay, student? 2 alpha chain and the 2 beta chain. So this is about the conformation of the proteins. Okay, primary structure. In the primary structure, you can easily find which amino acid in the first position, second, or the third. Okay, so here you can see here in the starting, it will be always will be the N terminal. So this is the C terminal. This is the N terminal, C terminal. This is the N terminal and the C terminal. Where ammonia group, NH3 group is there, that will be the N terminal. Where CO minus is there, that is called as the C terminal. Okay. Fine. So then we'll talk about the protein in the next class. Some important proteins are there. So then here I want to tell student in the most abundant protein in the animal kingdom. So which is the most abundant protein in the animal kingdom? Can anyone tell me? Collagen. Collagen is most abundant in animal kingdom. Okay, in the animal kingdom. When it comes to the Rubisco, what is the function of the Rubisco, student? In the Calvin cycle, you have learned about the Rubisco, right? It is the most abundant. It is the most abundant in biosphere. In the biosphere. In the biosphere, they ask means Rubisco. In the animal kingdom, they ask means collagen. Okay. So this is the end of the lecture. If you have any questions, you can ask me, or you can leave the class. Thank you so much. In the next class, we are going to study about the lipids and the enzymes. Okay, lipids, enzymes, and the nucleic acid. We will cover in the next class. Yeah. Thank you so much.